guys, good morning. Good morning, sir. We are from group 6 that we want to present about our discuss. There is a critical book report. But before we start it, let us introduce our group. Start from me. My name is Esther Lina Burusaragi and with my partner Ferawati Butar-Butar. So, with our title of the CBR, that is a developmental mechanism of evolution change. So, the first will be explained by me with our outline. The first, unity of type and condition of existence by Charles Darwin synthesis, and the second, precondition for evolution through developmental change, deep homology. And the last, mechanism of evolutionary change. So the first is the unity of type and condition of existence, Charles Darwin synthesis. So, in the 19th century, debates over the origin of species pit ways of viewing nature against each other. One view, championed by George Cuvier and Charles Bell, focus on the difference among species that allow species to adapt to its environment. Thus, the hand of the human, the flavor of the seal, and the wings of bird and bats were seen as marvelous contrivances. It's fashioned by the creator to allow this animal to adapt their condition of existence. And the other view, with the result by Etienne Hillary and Richard Owen, was that unity of type and Darwin is mixed to the component, where Darwin acknowledges the depth to these early debates when he wrote in 1859 that it's generally acknowledged that all organics have been formed on to great law. There is a unity of type and the condition of existence. Darwin went on to explain that theory will explain unity of the type but descent from a common ancestor. That is the change with the concept descends with modification, homologies between the embryonic and larva structure of the different phyla provided excellent. So, the next is about the precondition for evolution through the developmental change. The first, I will tell you about the modularity. This is the divergence through dissociation. So the first, the ability of one module to develop differently from other module, it's called the dissociation, where the modularity unit allows certain part of the body to change without interfering with the function of the other part. And you must know one of the most important discovery of evolutionary developmental biology is not only are the anatomical units modular, like the example that the one part of the body can develop differently than the other. But as you know, the DNA region that from the enhancer of gene are modular. This modularity of enhancer element allow particular set of gene to be activated together and permit a particular gene to become expressed in the several discrete places. Thus, if a particular gene loses or gains a modular enhancer element, the organism containing that particular allele will express the gene in the different place or at the different time that those organisms retaining the original allele. And with the result, the development of different anatomical and physiological morphologies. The next about this, that Davy blood group substance, where the V contain protein we can found in the Purkinje neuron, fan, and erythrocyte. So, uh, enhancer modularity has been known from the studies of the selectable traits in human population, where that is the Plasmodium vivax is a protozoan parasite that causes about the 75 million cases of the malaria each year. This form of malaria is not as lethal as falciparum malaria, but it can be incapacitating, causing severe pain, diarrhea, and fever. And some African populations are immune to PVVAX because the red blood cells lack protein. The Duffy glycoprotein that PVVAX needs to attach itself to the host red blood cell. And the lack of the Duffy glycoprotein was selected caused resistance to Vivax malaria, given that Plasmodium uses the protein infect the erythrocyte and probably is the one of the receptor of internucleon egg, or with the code EL8. This paracrine factor involved in the circulatory system in cerebellum. Also, the next about the PITX1 and stickable evolution. So, this is research in the analysis occur in the tree spine Stickleback fish, or the name of fish is Gastreutus aculeatus, where the freshwater. So the evolution is different is the freshwater and in the marine. But before that, freshwater stickable fish evolved from marine sticklebacks about the occur in the 12,000 years ago, when marine population colonized the newly from freshwater at the end of the last ice age. So I will explain to you about the picture. You can see. Yes, about the picture explain that modularity of development enhancer loss of the PITX1 gene expression in the pelvic region of the freshwater population of the tree spine stickleback. Okay, body plates and pelvic spine characterize marine population of this species. In the picture A, this is in the freshwater population, and the B pelvic spines are absent, as is most of the bony armor in imaginal ventral view of the embryo, where in situ hybridization rival pit at once expressed in the purple color in the pelvic area, as well as in sensory neuron, thymic cell, and nasal region of the marine population. The staining the pelvic region is absent in freshwater population, although is still seen in the other area. Okay, and the picture number 3, there is a model of evolution of pelvic spine loss for the enhancer region as postulated to reside near the PIT1 coding region. This enhancer direct to expression of the gene is the thymus, pelvic spine, sensory neuron, and nasal pit respectively. In population in the fresh water of the tree spine stickleback, the pelvic spine on hind limb enhancer model has been mutated, and the PITX1 genes fail to function this. Okay, this is about the picture. And the next, about the molecular parsimony, a gene duplication and divergence. And as you know, that we are explained to you before about the critical journal review is about the Hox gene, and it's important to know of you because it's the part of the evolution, and this book explain it about the molecular parsimony ini itu sangat berkaitan dengan Hox gene. Oke, langsung this is the Hox gene. Okay, the molecular parsimony ini it's some called toolkit. Toolkit is mean same as the molecular parsimony. That's developed from the lineage to lineage. Use the transcriptional factor like specs and Hox gene are found in the animal phyla that I said to you before. And the next that we know about the Hox gene, I will say to you before that is appear to specify the anterior and posterior body axis, even though the way these genes are activated differs enormously among nematodes, flies, and birds. Similarity, homologous or X specify head formation in both vertebrate and invertebrate. That the Hox gene ini occur that I say to you this occur in the Drosophila melanogaster and mouse. With the duplication and divergence, it's in the Hox gene we can see that in the globin genes and the collagen genes and the distallase genes, its member of the gene family is homologous to the other called paralogous. The example is the Athenapedia genes of the Drosophila in paralogu of the Drosophila ultrabithorax. There is a example picture about the type of that the first change in the Hox gene protein response element to downstream genes. The second picture is the change in Hox genes transcription pattern with a portion of the body, and the third change in Hox gene in transcription pattern between portion of the body, and the fourth is the butterfly change regulation or function of the downstream genes, change in the number of Hox gene. Okay, the next is about deep homology. Deep homology is the homologous pathway made of the homologous part are used for the same function in both protostomes and deuterostomes. Okay, as you can see the picture is about the Hox genes too. In 1940, Richard Goldsmith wrote that the accumulation of small genetic changes wasn't sufficient to generate evolutionary novel structure such as the neural crest, teeth, turtle, cells, feeder, or cnidocyte. He claimed that such evolution could occur only through inheritable changes in the genes that regulated development. This idea was brought up again in 1975 when Mary Clara King and Alan Wilson published a paper with the title Evolution at Two Levels in Human and Chimpanzees. This study showed that despite the large anatomical differences between chimpanzees and humans, their DNA was almost identical. The differences would be found in the regulatory genes that act during the development. With their said the organismal difference between chimpanzees and human would result chiefly from genetic change in a few regulatory systems, while amino acid substitution in general would rarely be a key factor in measure, adaptive, and safe. So, in the other word, the allelic substitution of the genes that encode protein sequence, which seems to be pretty much the same of chimpanzees and humans, were not seen as being important, and the important differences are where, when, and how much the genes are activated. In the 1977, the idea that change within regulatory genes is critical to evolution was extended by Francois Job, the Nobel laureate, who helped establish the operant model of gene regulation. And then, also in the 2004, 2004, has cataloged for ways in which Jacob tinkering can take place at the level of gene expression to generate phenotypic variation available for the natural selection. This is the first heterotopy. It's change in the location, and the second heterochrony is change in time, and the third heterometry, change in amount, and the last heterotypy, change in kind. You must remember it. And this book explains about the mechanism of evolutionary change is the for this type. Okay, so the first heterotopy. So sorry, it's change in location. Evolutionary change in the spatial arrangement of an animal embryonic development, complementary to heterochrony, a change to rate or timing of a development process. It was first identified by Ernest Haeckel in the 1866 and has remained less well studied than heterochrony. The second is the heterochrony or change in time. Heterochrony is any genetically frontal difference in the timing or duration of the developmental process in the organism compared to its ancestors or other organisms. This leads to change in the size, shape, characteristics, and even presence of certain organ and feature. Okay, you can see the picture. I will show you more big. That is a heterotopy, and it's the second picture. It's about the heterochrony. Okay. Right. Next, the third is the heterometry. It's change in amount. So this is the picture, right? So the heterometry is the one of the best example of the involved Darwin's finch, a set of closely related birds on the Galapagos Island. There are differences in the beak morphology between cactus fins, which have long narrow beaks helpful for probing cactus flowers, and ground fins with deep root beaks, which help them to crack open the seeds. And Abshanov et al. Found a correlation between beak shape and amount and timing of the BM4 expression. The same group also found drug microarray analyzed that calmodulin is expressed 15-fold higher in the beaks of cactus fins. And the correlation in the left picture is the correlation between back shape and the expression of BMP4 in 5 species Darwin finch, and the picture in the right is the correlation between beak length and the amount of calmodulin or CAM gene expression in 6 species Darwin finch. Okay, the last is the heterotypy, change in time. In heterochrony, heterometry, and heterotropy, Mutation affect the regulatory region of the genes. The gene product to protein remains the same. The change in heterotypy affect the coding region of the gene. So this is the picture about the graphic, yeah, and the graphic about how pregnancy have evolved in mammals. And as you know, one amazing feature of a mammal is the female uterus, which can nurture and protect a developing fetus within the mother's body, and the one key protein enabling this prolactin as is promote differentiation of uterine epithelial cell, regulate trophoblast growth, and down-regulate immune in the inflammatory response. And the mammalian HOXA11 appears to have undergone extensive mutation and selection in the lineage, giving rise to placental mammals such that it no association with a FOXO1A. This association allow the expression of the prolactin from a uterine specific enhancer, and the HOXA11 from opossum and the platypus and from chicks does not upregulate prolactin ability. In the picture explain of the ability the mammal HOXA protein in combination with FOXA1A to promote expression of the uterine prolactin enhancer. And for the addition, these changes can only be accomplished if the gene expression patterns are modular. That is, if they are controlled by different enhancer element, the modularity of developmental allows one part of the organism to change without the necessarily affecting the other parts. Okay, I think I'll explain until this. For the next explanation will be explained by my partner Vera. This occur in Drosophila and mouse. Okay, the explanation we can see.
Okay, my name is Vera. I will continue the next topic about developmental constraints on evolution. So, the first thing that you must know is the basic about what is developmental constraint. So, basically, there are only about 3 dozen major animal lineage, and they encompasses all the different body plans seen in the animal kingdom. So the question is, why don't we see more body plans among the living animals? So to this question, the answer is we must consider the constraints that development imposes on evolution. In the living animals, the possible phenotypes that can be created are limited by the interaction that are possible among the molecules and between the molecules. So, this interaction of molecules is the one that allow change to occur in certain directions more easily than other factors. Nah, sekarang kita akan membahas apa sebenarnya developmental constraints itu berarti. So, the developmental constraints are the restraints on phenotype production. Dalam evolusi sendiri, constraints ini akan dibagi ke dalam kategori utama, yakni yang pertama physical, the second is morphogenetic, and the last is phyletic. Okay, the next is the physical constraints. In physical constraints, the law of diffusion, hydrology, and physical support allow only certain mechanisms of development to occur. So, in physical constraints, you must remember that only certain mechanisms of development that occur the diffusion, hydraulic, and the physical support. For example, the first one is the structural parameters and fluid dynamics forbid that existence on 5-foot-tall mosquitos. And the second example is in Drosophila sperm, the type of tubulin that can be used in the axoneme is constrained by the need for certain physical properties. The second is the morphogenetic constraints, the reaction diffusion model. In these constraints, when organisms depart from their normal development, they do so in only limited number of ways. For the example, if a longer limbs is favorable in a given environment, the humerus may become elongated, but one never sees two smaller humerus or humeri joined together in tandem, although one could imagine that the selective advantage that such an arrangement might have. This observation indicates a construction scheme that has certain rules. Jadi pada dasarnya, kita dapat melihat contoh yang dikatakan bahwa humerus belum pernah ditemukan tumbuh atau bergabung pada satu tandem. Hanya ada humerus yang memanjang, namun tidak ada dua humerus sekaligus yang bergabung bersama. Nah, untuk itu akan kita bahas pada selanjutnya. Jadi, selain dari humerus yang tidak pernah tumbuh barengan pada satu tandem tadi itu, ada hal-hal lainnya yang juga tidak pernah terlihat, seperti middle digits that never shorter than surrounding digits. Jadi tidak pernah jari tengah itu lebih pendek dari jari-jari lainnya. Hal inilah yang membangun rasa penasaran oleh Bateson and Albert. So, after observing this phenomena, Bateson in 1894 and Albert in 1989 noted that when organisms depart from their normal development, they do so in only a limited number of ways. The next person that being involved is Alan Turing. So in 1952, Alan Turing created the very first reaction diffusion model for the developmental patterning. He proposed a model wherein two homogeneously distributed substances, which is substance P and substance S, interact to produce the stable patterns during the morphogenesis. And these patterns would represent the regional differences in concentrating of the two. And the Turing's mathematics already been used to model the spiral patterning of slime molds and the polar organization of the limb, also the pigment patterns of mammal, fish, and the snails. Okay, we can see this figure now. This is one of the Turing's model of pattern generation. In this picture, we can observe in the A is generation of periodic partial heterogeneity can come about spontaneously when the two reactants, the S and the P, are mixed together under the condition that S inhibits P and P catalyzes the production of both S and P, and then S diffuse faster than P. This condition yields a peak of P and a lower peak of S at the same place. And for the next picture is the distribution of the reactants is initially random, and their concentration fluctuate over a given average. As P increases locally, it produces more S, which is diffuses to inhibit more peaks of P from forming in the vicinity of its production, and the result is a series of P peaks at the regular intervals. Okay, the next topic is the evolution of mammal's teeth. In this picture, we can see the GIS analysis of gene activity in the formation of the first set of cups in mouse and foal molars. We can see the different shape between the teeth of mouse and foal and how the gene actively forming the teeth of the organisms. And in 2004 and 2010, Slaughter-Cudit and Zernfall showed how the reaction diffusion model can explain the differences in gene expression between mice and foals. In these pictures, you can see that mammalian tooth cups pattern modeled by the reaction diffusion equation. In these pictures are seen that the reaction diffusion mechanism serves as a motor regulating the genes responsible for slowing the growth of the enamel-forming cells and promoting the proliferation of the dentin pulp-forming cells, also showing that possible formation of the four-cups tooth of Hyracotherium, which is a mammal that may be earlier member of the horse lineage. The next is the third constraint called pietic constraints. In pietic constraints is more focused in historical restriction based on the genetics of an organism's development. For example, inductive interaction generates structure. The first one, the notochord, is vestigial in adult vertebrate but functional in the specification of the neural tube. And the second, pronepros of cheek, is the source of uretic bud that induce the formation of functional kidney. The next is selectable epigenetic fetiation. In this topic, I have two important points that explain about the selectable epigenetic fetiation. The first one, developmental models can include several tissue types such the correlated progression occurs. In this point, here a change in one portion of the model that causes changes in the other portion. For the example, when the skeletal bones change, the nerves and also the muscles that serving them, the skeletal bones that already change, also change at the same time. So, the skeletal bones that already change, affecting the nerves and the muscles to be changed either. The second point, tissue interaction have to be conserved, and if one component changes, the other must change either. And for the example, if a ligand changes, its receptor must change, and the reproductive isolation may result from the changes in sperm or the egg protein. In this slide, we can see the picture of epigenetic forms of tooth flax. In picture A, the typical linearia with a relatively unmethylated cicolin gene. That was the epigenetic forms in picture A, and while the picture B is the cochlidea gene of the Peloria variant is relatively heavily methylated. The epialleles that create the different phenotypes of the species are stably inherited. The next topic is HSP90 and genetic assimilation. What is HSP90? HSP is heat shock protein. That means the protein that binds to a set of signal transduction molecules that are inherently unstable. This provides a way to resist fluctuation due to slight mutation or environmental change, and also responsible for allowing mutation to accumulate by keeping them from being expressed until the environment changes. And for noted, transient decrease in HSP90 or damage would uncover the pre-existing genetic interaction that would produce the morphological variations. So this protein really have an important roles in genetic assimilation that we already know. It has several roles that really needed in producing the morphological variations. In this slide, we can see the picture of the phenocopy of the bithorax mutation in Drosophila. In picture A is a bithorax phenotype produced after the treatment of the embryo with ether, and the forewings have been removed to show the aberrant metathorax. This particular individual is actually from the assimilated stock that produce this phenotype without being exposed to ether. And in the picture B is the selection experiments for a or against the Bithorax-like response to ether treatment. Two experiments are shown, which in both cases, one group was selected for the trait and the other group was selected against the trait. The last topic, a developmental account of evolution. The developmental biology has provided the underpinning of variation modulatory, molecular parsimony, gene without destroying an organisms. The developmental biology also has uncovered four modes of genetic changes that can produce new and large variations. So, the developmental genetic model has been formulated to account for both the homologies seen in evolution and the differences. But we know that we are still approaching evolution in the two ways that Darwin recognized, and descent with the modification remains central. But at some point, now we can answer some evolutionary questions using both population genetics and developmental genetics. Thus, we can say that evolution is generated by heritable changes in development. By integrating the population genetics with developmental genetics, and also the embryology, we can now begin to explain the construction and evolution of biodiversity. Finally, we come to the end of this presentation of critical book report from Group 6 about the developmental mechanisms of evolutionary change. We say thank you for all your attention, and also we do apologize for our mistakes in words, also pronunciation. That's all from Group 6. Thank you.